Hello and welcome to my talk. My name is Suarez-Rodriguez and I am going to talk about a study where we explore the implications of a physical pollutant. All environments are changing through time, and since human populations have expanded so much, these additional costs have been showing consequences. Human activities are part of the most abrupt, continuous and irreversible modification of habitats. In particular, pollution has played a big role in the changes organisms have been going through for the last centuries. Chemical pollution has been one of the most popular types of pollution because now we know that almost all chemicals in the materials we use interfere in some way with wildlife. However, the physical presence of objects can also interact with the species. And the simplest way of thinking this is that there is a special distribution of biotic and abiotic factors in a given moment of an environment. This change in the external materials, the discharge of any materials, can modify the physical disposition of boundaries, landmarks, and territories of these environments. And for example, in this work with this convict title, it was found that when incorporating artificial landmarks, they use cans, their territorial behavior will change. So fish move their territories' boundaries, making them smaller but easy for them to defend. Plastics have been very important to human civilization development. It's one of the most common materials we use. And in addition, they are almost indestructible. So they have become a major problem. And most studies about chemicals implied in plastics have shown effects on different systems in different organisms. But in this study, I want to talk of what could happen if plastics are just physically present in an environment. We developed this question of how different presentations of the same pollutant, for example plastic bags, affect behavior of zebrafish. For this, I used adult zebrafish and divided them in mixed groups of 10 and 20 fish. Then I set up 5 tanks and assigned each of them to a different treatment. These treatments were different from each other because of the form of the materials. So it could be in a continuous material or as a debris, mimicking a breaking bag. And then this could be floating or sinking at the bottom of the tank. I film and analyze videos where I look at activity levels and social behavior. All my statistical analyses are based on selecting the best models through AICC. As my first measurement of activity levels, I analyzed latency to feed. Here the latency in the y-axis was better explained by the group size. Larger groups approached faster to the food than smaller groups. My second measurement of activity was the distance moved that was affected by the treatment and the group size. Here, the groups from the bottom debris treatment moved less than any other treatment, and also the smaller groups moved more distance. For social behavior, the first thing I measured was the proximity, which is the time individuals spend near each other. And this varies by group size, where groups of 10 were in proximity of others for more time than groups of 20. As my second measurement of group behavior, I analyzed the relative movement of time the fish spent moving to others. This variable was best explained by the treatment. Here the presence of the material as debris made the fish spend less time moving near each other compared with treatments that had the plastic bags as continuous material. Larger groups were bolder approaching to the food faster, and this behavior has been associated with the perception of risk in fish. In this case, it is likely that larger groups were bolder because feeling safe. Also, they didn't move as much distance. So this could be related to what is thought as better spreading of the information from the surroundings in larger groups, giving the information of predators and food faster, so they didn't have to move as much as small groups. What is interesting is that they spend less time in proximity. However, this could be a result of looser cohesion between individuals that we need to explore. The form of the material affected the relative movement to other individuals, and it is possible that when the material is in patches, they are exploring more the objects. As it has been noted in the lab, zebrafish territorial behavior escalates when there are objects to defend. They also make them aggressive, so they keep apart from each other more. But we still need to evaluate videos. The position of the material also showed effects on behavior of zebrafish, and in particular, when the material was continuous at the top. It was obvious that they were using it as a refuge. We did not detect that this treatment was the one where fish move less distance, but it is one of the 2 treatments with less activities. When fish explored the empty space, they tended to do it in groups, so this might reflect that leaving the refuge was taken cautiously. I think what we can take from this study is that we need to start targeting different aspects of the garbage we have in the environments and analyze the consequences. In this case, behavior allows us to detect how different groups of fish react to that change in the physical environment, even when they were experiencing the same external material. And that large materials of garbage can impose other pressures, not only because of their chemical composition, but for the physical presence in an environment. So with this, I want to thank the Martins Lab for their support in this study, to you for listening, and I am open to any questions.